Ready? You can see it. Hello.
Yes, sir.
Hello. Okay. So I think all the syllabus is covered as per your syllabus, but I explain elaborately in the different topics in the different day also. Okay. Just elaborately discuss with this topic. Antibacterial chemicals and the bactericidal and bacteriostatic agents. Those are the bactericidal and bacteriostatic. There is a well defined between the bactericidal and the bacteriostatic agents. Those disinfectant or the antibiotics which are bacteriostatics and which are bactericidal and which effect is needed, that is very important for human as well as the living things. So we can discuss about this. First is the antibacterial, those agents which inhibit the growth of the or kill the microbes or bacteria especially, that is called the antibacterial agent. The antibacterial agent, those agents which are inhibit or growth of the microorganism, their classification. And what is bactericidals? That means those agents which totally kill the microorganism, that is called the bactericidal. And bacteriostatic means to arrest the growth or inhibit the growth of the bacteria is called the bacteriostatic. And their mechanisms, bactericidal and the bacteriostatic, the microorganism and how it will be act, their mechanism will be discussed in these chapters. What are the advantage as well as the disadvantage of the bacteriostatic as well as the bactericidal agents? The antibacterials. Antibacterials is an agent that interfere with the growth and reproduction of the bacteria. The agent which interfere with the growth and reproduction. That means their growth will be hampered and their reproduction. So bacteria cannot produce in the another bacteria. That is called the antibacterial agent. And these agents kill or prevent the bacterial infection by fighting against the bacteria. Basically, these bacterial agents, the bacteria infect into the body and that produce infection, and that will be fight against the bacteria. And heat chemicals such as chlorine and all the antibiotics drugs are having the antibacterial properties. Not only the antibiotics, but also the heat, some physical agents and the chemical agents such as the chlorine and others material, which also the which have the chemicals and the others chemical also that having the same antibacterial action. Antibiotics and the antibacterial both attack the bacteria. Not only the antibiotics the special kinds of antibacterial agents and the antibacterial especially to prevent against the growth of the bacteria, both are attack the bacteria. Antibiotic also known as the antibacterial agent, but all antibacterial agents are not antibiotics. But antibiotic means so many compounds that will be not only inhibit the growth of the bacteria but also the other organisms like fungi, mold and viral etc. also. These antibacterial can be treated as a viral infection like the flu and the cold. This is the antibacterial agent. And antibiotics means for the inhibit the growth of the microorganism, especially for the bacteria. And these are the same essential related terms starting of the antibacterial agent that are motive. So be careful these terms. What is the biocides? That means a widespread and chemical and physical agent which inactivate the microorganism. That is called the biocides. That means biocide, bio means lives and the sides means kill. That means that type of microorganism, it is a widespread chemical and physical agent which inactive the microorganisms. And bacteriostatic is a property of the specific biocides agents which is able to bacterial multiplication. Bacteriostatic means the property of the specific biocides agents which able to bacterial multiplication, which are able to inhibit the bacterial multiplication. And bactericidal, it is a biotic biocides able to kill the bacteria. Only the bacteriostatic, there is only difference static, that means the growth will be inhibited. But in case of bactericidal, it is kill the bacteria. And what are the disinfectant? Basically, it is used in the inanimate object and to reduce only viable microorganism on the inanimate object. That means not living organisms. That is called the disinfectant. What are septic? It is characterized by the response of a pathology characterized in the presence of the pathogenic microorganism in the living tissue. In case of human or other things, in case of living tissue, it introduce some organisms and that produce some infection. That is called the septic. And antiseptics, which is inhibit the growth of the microorganisms, inhibit the growth of the microorganism is in or invading the tissue. And aseptic, to keep free from microorganism, you are used in the microbiology labs for the aseptic technique. Not practical, but then theoretically you know what is aseptic technique. If you did not know, next further class I discuss about the what are the aseptic technique and what are the aseptic area and how it will be maintained to basically for the preparation of the ophthalmic and parenteral preparation in the aseptic area. Antibiotics, it is a natural occurring and synthetic organic compound which is inhibit or destroy the bacteria at low concentration. That is called antibiotics. Nowadays there's so many antibiotics, and this is the highest most used in the field of medicine as well as the misuse of the medicine. Antibiotics generally it was produced in the naturally or by the synthetic procedure from the one microorganisms, and they are used or used against the another microorganism and had killed or the static the or growth inhibited at low concentration. That is called the minimum inhibitory concentration. That is called the antibiotics. So these are the very important terms. We remember it in case of microbiology, not all the microbiology, in case of the pharmaceuticals also. So be careful, be take care, be learn about it and remember it. These are the terms. The antibacterial agents, some antibacterial agents here, only the soaps. The generally we are use the soaps in the corona COVID to always in the news or the newsletter or there's so many others informal medical bulletin. They can use the soaps, hand soaps. Sorry, use for every 30-minute interval for 10 seconds to inhibit the growth or to remove the virus. So soap is a one antibacterial, potent antibacterial agent to inhibit the growth or remove the microorganisms. Another is the hand lotion. Sometimes we are use some hand lotion nowadays. At present, you are very aware about this hand lotion. And disinfectant also same, the Lysol, Dettol, chlorgenilol, and the surface spray. Sometimes the agent surface can be removed spray by the ethyl alcohol or by the disinfectants, and the mouthwashes also. These are also sometimes there's a bad smell coming from your mouth. At that time, you can use some mouthwashes. Generally, the chlorhexidine types of antibacterial agent or antimicrobial agent or citridine, citramide. So that type of mouthwash which are used as a disinfectant and to kill the microorganism or to eradicate the microorganism. Another is the toothpaste. Obviously, you are used in the daily life, and the toothpaste for especially for the cleansing and microbial free of the teeth. Another is the antibiotics. Antibiotics and are the most important and the potent antimicrobial agents which are used for the inhibit or kill the microorganisms. Next, the classification and the groups are based on these types, antibacterial agent and the subclass of antibiotics, and can be classified into the major 5 groups. One is the type of action. As this antibacterial agent or especially the antibiotics which are classified based on the type of action, which type of action, which act on the which type of action, where it will be act, where, and next is the based on the source, how it will be collected and how it will be formed. And number 3 is the spectrum of activity. It is a broad spectrum or narrow spectrum. Number 4, it is a chemical structure. Obviously, what is the chemical structure? Based on the chemical structure, They are all both different classified, and they are broadly classified in the based on their chemical structure. Different antibiotics having their different chemical structure. And finally, the function. Obviously, the function is the most important parameter that can be based on this parameter. You can classify the antibacterial, especially the antibiotic, where it will be, it is antibacterial, antifungal, or antiviral, or antiprotozoal, or other drugs. On the type of vaccine, this is the bactericidal, and the bacteriostatic. Bacteriostatic, broadly it will be classified in the type of action. It will be two types: one the bacteriocidal, another is the bacteriostatic. Bactericidal means to kill the microorganism or the bacteria, and in case of bacteriostatic, to inhibit or growth the arrest of the bacteria. Bactericidal, those antibacterial agents actually kill the bacteria by any mechanism depending on the antibiotics. Those antibiotics that actually kill the bacteria by any mechanisms depending on the antibiotics, it will be classified into the depend on the, it inhibit the cell wall synthesis inhibitor. Cell wall is a very important component for the bacteria as well as the microorganisms. Some microorganisms, the outer covering is called the cell wall, which is composed by the peptidoglycan, and if the cell wall is ruptured, suppose your room wall will be break down, the room will be break. So at that types of, if the living system is covered by the cell wall, if the cell wall ruptured or inhibited, that immediately the bacteria or the microorganism can be killed. So cell wall inhibitor, cell wall synthesis inhibitor, one types of antibiotics. Action is the irreversible and do not work with the immune system of the host. Obviously, this antibiotic generally, the or ideal characteristic of the antibiotic, generally it killed or the kill the microorganisms or arrest the growth of the only the microbes, not in the host cell. So for example, in case of antibiotic used in the humans, human host cannot be affected, only the microorganism can be affected. And minimum bactericidal concentration drug is required for kill 99.99% of the bacterial population. Minimum effective one. Suppose one drug 100 mg not kill the microorganism, 500 milligram it kill the microorganism, but 600 kill the microorganism, 1000 milligram kill the microorganism. Which concentration? The minimum effective concentration. That means here is the 500 milligram is the minimum effective concentration, because if high concentration is used, though the therapeutic activity will be increased, simultaneously their toxicity will be increased. So at that concentration, what is the minimum concentration? That concentration microorganism will not produce or kill or invade the microorganism. So here is the most least concentration where the microorganism will not grow. That types of concentration can use. Example, and the beta-lactam antibiotic in this group of the penicillin, cephalosporin, and other, and daptosomycin, aminoglycoside like streptomycin, gentamicin, clindamycin, metronidazole, fluoroquinolone, ciprofloxacin, ofloxacin, levofloxacin, parfloxacin, and vancomycin. These are the types of drugs which are used for this bactericidal action. What is the bacteriostatic? Those antibiotics that stops the inhibit the growth of the bacteria, and that means the multiplication and generation of the bacteria, but they do not kill the bacteria. It is called the bacteriostatic. Static one is static, not totally killed. And slow growth and reproduction. Sometimes the growth rate is slow and the reproduction will be slow. That is called the bacteriostatic. And inhibit the DNA replication, protein synthesis of the bacteria. Basically inhibit, not kill the bacteria. Inhibit the growth replication and the protein synthesis. Ultimately their growth will be arrested. And work with the immune system of the host. Work with the immune, here the work system is work with the immune system of the host, and minimum drug concentration is required. Same criteria. For example, the tetracycline, penicillin, sulfonamides, legionnaires, chloramphenicol, clindamycin, and the macrolides group of antibiotic under this class, erythromycin, azithromycin, and clarithromycin, which are under the class of macrolides antibiotics. So these types of antibiotics which are used as a bacteriostatic. What are the mechanism action? Number one, the inhibition and target of the bacterial cell wall biosynthesis inhibitor. This is the one of the most important as well as the fastest antibiotics which are used for the cell wall synthesis inhibitor, because you know in your bacteriology by the manosphere, what is the composition of the cell wall? The basic composition of the cell wall, basic component is the peptidoglycan in case of gram-positive bacteria and lipopolysaccharide in case of gram-negative bacteria. So cell wall composition is very clear crystal idea, and that cell wall is to protect the bacteria and to survive of the bacteria. But when this antibiotic agent or antibiotics used, and those antibiotics which are inhibit the cell wall synthesis, that is called the cell wall synthesis inhibitor, basically the beta-lactam antibiotics. Here for example, the penicillin, not only penicillin group of antibiotic, penicillin, amoxicillin, methyl penicillin. So these are the types of groups, and another is the cephalosporin group of, and cephalosporin, cephaloxin, cephaloridin, cephalosporin. So these are the basically the beta-lactam group of antibiotics, penicillin and cephalosporin, to inhibit the cell wall synthesis inhibition and the target of the bacterial protein biosynthesis. Another is the, your protein is the very important molecule to growth of the bacteria, not only the bacteria but other also. But here the protein synthesis biosynthesis can be prevented, and generally protein is synthesized mechanism by the two subunit. One is the by the two basically protein synthesis in the factory house, where it is called the ribosome, and the ribosome which consists of the two. In case of prokaryotic, one is the 30S, another is the 50S, and the total is the 70S. It is a mystery, and there's some mysterious and some interesting thing is the 30S and the 50S, but the total ribosomal subunit is called 70S, and inhibit target inhibition and the target with the bacterial nucleic acid. This is also another important for the not only the reproductive system but also other characteristic of the microorganism that will be dependent upon the nucleic acid or nucleic acid synthesis, the DNA or sometimes the RNA. If it is inhibited, immediately the bacteria will be killed, and target the bacterial cell membrane function also. Inside the bacterial cell wall, some are the cell membrane. The cell membrane will be destroyed, and ultimately the inhibit and growth of the microorganism. And inhibition of the metabolic pathway. Sometimes the microorganism, there's some metabolic pathway, and some steps can be arrested. Or in case of, for example, the sulfonamides, generally there's metabolic pathway, folic acid to folinic acid, it will be prevented, or dihydrofolate reductase or tetrahydrofolate reductase can be inhibited. And ultimately, the folic acid cannot be synthesized. So their metabolic pathway will be hampered due to the use of some antibiotics. And by this mechanism, so many drugs can act on this to kill the microorganisms. Another is the inhibition of the ATP synthesis. ATP is the one of the most important energy molecule for the growth, metabolic process, and everything. ATP is needed some. So if the ATP can be synthesized, can be prevented, then immediately the microorganism will be weak, and ultimately the growth will be arrested, and finally the death may be occurs. The bactericidals and the bacteriostatic agent, inhibition and target of the bacterial cell wall or bacterial cell wall biosynthesis. Target the bacterial cell membrane function, and inhibition target of the bacterial protein biosynthesis, inhibition of the bacterial nucleic acid synthesis. All these are the four class which are acts as a bactericidal action. And what are the bacteriostatic? Especially by this mechanism, inhibit of the metabolic pathway. That means metabolic pathway or inhibit ATP synthesis, inhibits to target the nucleic acid synthesis and protein synthesis. So in case of protein and nucleic acid, these are the two common pathway. Either it is, sometimes it will act as a bactericidal or sometimes bacteriostatic. But why bactericidal? When it is bactericidal and when it is bacteriostatic, it is depend upon the especially microorganism, which microorganism or which antibiotic used for the which type of microorganisms. Especially the depend upon the variety of the microorganism. If it is killed, then it is bactericidal. If not killed by the mechanism nucleic acid synthesis or the protein synthesis, if it is not killed, that is called bactericidal. So these are the very good picture. It will be, you can say that it is the cell wall, the beta-lactam in the cell wall, because it is the outer wall of the microorganisms, and which is essential for the prevention of the all the materials of the microorganisms. And the cell wall is very important compound, and if it is ruptured or it will be destroyed by any means, the microorganism immediately killed. So by the beta lactam types of antibiotic, penicillin, cephalosporin, monobactam, carbapenem, and glycopeptides, vancomycin and bacillin. Those are the antibiotics which are inhibit the bacterial cell wall. Bacterial cell wall. Number two, DNA synthesis. DNA, obviously you know the DNA is the most important parameter or most important parameter for the synthesis of the protein. Not only the protein, all the physical and chemical character of the my living body can be depend upon this DNA. So DNA synthesis can be inhibited by the fluoroquinolones like ciprofloxacin, levofloxacin, moxifloxacin. RNA synthesis like rifampin, and acyclovir also. The plasma membrane. Sometimes the plasma membrane they got just inside the cell wall. It is called the plasma membrane. And polymyxin B, colistin, lipopeptides, and daptomycin and cytoplasm, which are used for the inhibit the cell membrane. Ribosome, there is the 30s Rahab unit, aminoglycoside, tetracycline, and 50s ribosome for the for 50s and 30s ribosome. And 30s the macrolides, antibiotics, chloramphenicol, oxytocin, and metabolic pathway by the synthesis of sulfonamides and hello, huh, morning bolo, huh. Okay.